So welcome back from the break. Let's take a look at our concept check question so we can move on to the next supergroup. Why are the mitochondria of diplomonads and parabasalids often described as highly reduced? Remember that these are mitochondria that no longer carry out aerobic respiration. They can, however, carry out something analogous to partial function of the mitochondria. But largely, these cells rely on anaerobic respiration, glycolysis in the cytoplasm. But these are remnants of mitochondria. And we know that there were once fully functional mitochondria there because the mitochondrial gene sequences that are found in the nuclear DNA of all other eukaryotes are also found in the nuclear DNA of these cells. So we know that even though the mitochondria that are present now lack normal mitochondrial function, hence the term reduced, these were once fully functional mitochondria. So their function, whatever it is now no longer the function of typical mitochondria, hence the term reduced function. So the next supergroup is the SAR group. This is the largest of the four supergroups. This is also the one that for the moment is somewhat controversial, although it is becoming less controversial as a consequence of further gene analysis that suggests that these three groups really do belong together. The three components of the SAR clade really do belong together, at least at the genetic level. In terms of functionality and in terms of origins, that's a different story. And the reason why is because this group is characterized by the presence of both photosynthetic and non-photosynthetic representatives in all three clades, the stromenopials, the alveolates, and the rosarians. And in the case of eukaryotes that got their chloroplast through endosymbiosis of a cyanobacterium, the nucleus of the host cells contains gene sequences or genes derived from the symbiont, from the cyanobacterium through horizontal gene transfer. And so when you look at plants, when you look at algae, when you look at any protist that possesses a chloroplast as a consequence of the endosymbiosis of a chloroplast, of a cyanobacterium, there are chloroplast genes in the nucleus. Now, when you look at the non-photosynthetic individuals of all of these clades, the one thing that is missing are genes derived from chloroplasts or from plastids, which is the umbrella term that is used for the chloroplasts and other related organelles, leucoplasts and so forth. The genes that we would expect to find in the nucleus are missing. So that means that these non-photosynthetic members almost certainly did not evolve through loss of the chloroplast, as we've seen in the parabasalids and the diplomonads. If they lack the gene sequences, that suggests that they've never had chloroplasts. So what do these non-photosynthetic members represent? Are these descendants of the common ancestor of these clades that diverged from the other members of the clades that are photosynthetic before the capture of the algae that contributed the chloroplast? Or is there something else going on here? In terms of the phylogeny of this clade, evolutionarily, there is some controversy here. There is no question that in terms of the genes that have been sequenced from the genomic DNA, from the nuclear DNA, that these three clades do belong together in the supergroup. But the phylogeny of the non-photosynthetic versus photosynthetic members of each of the clades is open to question right now. That is the source of controversy within the cyclade. So let's take a look at the three groups. So the Stramenopila, or the Stramenopils, are so named because of two Latin words. Pilus is the Latin word for hair, and the Latin word for straw is stramen. So when you see the term Stramenopil, what it actually means is hairy straw, or in this case, a hairy flagellum. And that means that one of the two flagella, again, these are biflagellated, as all the protists are in 3 of the 4 supergroups where there are flagella present. One of the two flagella is hairy in appearance and the other is smooth in appearance. So you can see that here. Here's the hairy flagellum, here's the smooth flagellum. So Stramenopil, hairy flagella. And so this is a common characteristic amongst all the flagellated members of this clade. Again, they represent a single clade through genetic sequences, but they also have structural similarities. So the first members of the group are the diatoms. These are sometimes referred to as algae, but they are strictly not algae. You can see that I've used the reference unicellular algae because that's a terminology that a lot of people use, but these are strictly not algae in the stricter sense of the word. They are photosynthetic, however. And when you look at them, they have a very yellow appearance. I don't have the picture in here. I must have taken it out. They have a very yellow appearance because of the accessory pigments that they have. They also have a very characteristic shell around the outside that is made of a glassy material called hydrated silica. So silica is a reference to the glassy material. Glass is actually a silica material. Think sand. Glass is melted sand. Sand is the silica. So this is hydrated silica, meaning that it's wet silica material. And each diatom has a unique shell surrounding it, and it's made out of 2 parts that fit together very much like a petri dish, with one shell slightly overlapping the other shell. The shell has little holes in it, which allows for cytoplasm to emerge through this, so it can send out extensions. Not really pseudopodia, because pseudopodia are kind of chunky extensions from an amoeba, but it can do a similar sort of thing. There are several species, or there is a genus actually of diatoms. Sorry, I don't want to go there because I'm thinking about something else. So I'll retract that for now. I'm getting a little confused about which group I'm talking about. Yeah, I get confused by these guys as well. There's so much to deal with. So if you're finding yourself, if you've been reading the chapter, you're looking at the presentation now to try and make sense of something that you read in the chapter and you're still confused, welcome to the party. As much as I've been teaching this for many years, and as much as I've been trying to read up and make sure that I understand everything that's going on with all of this, it still gets confusing, and there can be some crossover. So diatoms. These are interesting for a number of different reasons, apart from the presence of these very characteristic glassy shells. One thing is that they can actually represent a really good way of removing carbon in the form of carbon dioxide from the atmosphere, because they can be found in both fresh water and in seawater. When they sink to the bottom of the bodies of water that they're living in, it can take a tremendous amount of time for the material of the cell to be decomposed by bacteria, and the subsequent carbon dioxide that is produced as a consequence of that decomposition through respiration remains in the water rather than entering the atmosphere because it's so deep in the bodies of water. Another thing that you may not recognize is that diatomaceous earth, which is the accumulations of the fossilized hydrated silica shells, serves a number of different purposes. And you have almost certainly utilized diatomaceous earth in some fashion today. So one way would be simply drinking water, because all water is purified in part by being filtered through a diatomaceous earth filter. All these little holes in the silica shells can filter out small organic particles and other particles almost down to the level of virus particles. It can certainly filter out bacteria. It can't quite get down to filtering out virus particles. So this serves as a really good filter bed for filtering water. And it's cheap because there are large deposits of diatomaceous earth around the world, indicating that at previous times in Earth's history, there have been substantial numbers of diatoms in water bodies. Also, diatomaceous earth is used as a mild abrasive in toothpaste. So if you brushed your teeth today, and you did, right? Then the mild abrasive effect of the toothpaste to remove buildup of deposits on your teeth is provided by diatomaceous earth in the toothpaste. Another use of diatomaceous earth for anyone who might be interested in pursuing a career in forensic science is that what we've discovered about the populations of diatoms in different bodies of fresh water, in different lakes and streams, is that each body of fresh water has its own unique population of diatoms. And these can be used diagnostically to determine where samples of water have come from, or where items that have been in fresh water may have been, as far as the freshwater body is concerned. If there's some question as to whether an item was in fresh water and which body of fresh water it was in, diatoms can actually be used diagnostically to identify which body of fresh water the item was in, or which body of fresh water the sample of water was taken from. Because, as I said, each body of fresh water has its own unique population of diatoms. And remember, we can identify each species of diatom because they have their own unique hydrated silica shell that is unique to each species. The shape of it is unique to each species. So these are really interesting organisms for a number of different reasons. Oh, one more use of diatomaceous earth. This one's a little bit more difficult to use or to understand immediately, but you can use diatomaceous earth as a non-toxic insecticide. And the reason why is because the hydrated silica, remember it's glass, has sharp edges. And if you sprinkle that down, when an insect walks through it, its cytoskeleton gets cut by the sharp edges of the diatomaceous earth, the diatom skeletons. And that causes the insect to dehydrate because it's lost its waterproof coating now, and it will quickly dehydrate and die. So if you don't like using chemicals as an insecticide, this is an alternative. And I know of at least one place here in Orlando that actually sells diatomaceous earth as an insecticide. I'm not sure how much it costs. I've never gone in to ask about it, but I thought that was kind of interesting use for diatomaceous earth. Of course, if you vacuum clean on a regular basis, you'd have to go back and relay the diatomaceous earth again to continue to keep the insects out of your carpet. Kind of interesting, I guess. Golden algae are one of the groups that are not mentioned in the textbook anymore. For some reason, they've decided to drop them, but these are also straminopeals. They have a reproductive cell called a zoocyte that is biflagellated and has a hairy flagellum as one of the two flagella. These belong to a group called Chrysophyta. The phyta tells you that it is photosynthetic. The diatoms, by the way, belong to a group called the Bacilliarophyta. You can understand why we don't necessarily want to use the proper clade name. We just use the term diatom. But chrysophyta are the golden algae. And this is because of the presence of carotenoids that give them a very characteristic yellow color. So here you can see the colony of chrysophyta. So a large number of golden algae are colonial, but the vast majority are unicellular. Here you can see the flagellated zoocyte and the flagellum, and then particularly the hairy flagellum on the reproductive cell. And that's all you need to know about these. Brown algae, or phaeophyta, are the largest group of algae, also the most complex, and also the first group of protists that we would identify as seaweeds. Now, what you need to recognize about the term seaweed is that it is not taxonomic. It has no place in phylogeny. It is a general term to describe the large multicellular algae that come from the phaeophyta, the brown algae, the red algae, or rhodophyta, and the green algae, or chlorophyta. Seaweeds are found in all three of these clades. And because all three of these clades contain multicellular large algae that have been termed seaweeds, the term seaweed cannot be considered to be taxonomic. It is just a descriptive term. Because when you say seaweed, most people know what you're talking about. The vast majority of brown algae are marine and live in the coastal waters. They need to be near the surface, in part because they carry out photosynthesis using chlorophyll A and chlorophyll B, and so they need to be relatively close to the surface because the wavelengths of light that they're dependent upon do not penetrate the sea surface particularly well. But that doesn't mean they can't get large. Some of the large seaweeds, like the kelps here, can be over 100 feet in length. They are anchored to the seafloor, but then they grow up to the surface and they'll have a part of the seaweed floating on the surface. And in fact, the body of the brown algae supports that. So here's a picture of a brown algae. It has three components: the holdfast, which is a very root-like structure that anchors it to the substratum, the stipe, which is very stem-like, and the blade, which is very leaf-like. But because these lack vascular tissue, you can't refer to them as roots, stems, and leaves. So we use these alternative names. Now, in the giant kelps that grow off the coast of California and elsewhere around the world, there are also floats associated with the stipe and the blades that allows it to hold itself upright and also to float on the surface. These are gas-filled structures that allow the structure of the algae to float in the water. What you may also not realize is that these have commercial value, particularly the brown algae that grow in what is known as the surf zone, where the waves are crashing. Because they get thrashed around by the waves, they need to have buffer material to prevent cellular damage. And so they have very complex polysaccharide material called mucopolysaccharide that acts as a buffer. And we utilize several of them. So we use a polysaccharide called algin that comes from brown algae. We use another one called agar, or agar, as some people pronounce it. That's the material that we use as support material for growing bacteria. And then there's another one called carrageenan. And if you've ever eaten low-fat dairy food like sour cream or a yogurt or an ice cream, anything that has low fat, but it still has a creamy texture, take a look at the ingredients, because almost certainly you're going to find carrageenan as a component. Because carrageenan feels to the tongue like fat. It has a tongue texture like fat. So when food manufacturers make a low-fat food, they want it still to have that smooth, creamy texture that we all like on our tongue. So they add the carrageenan in to provide that tough texture. And carrageenan is a polysaccharide, so it doesn't come calorie-free. And so consequently, some low-fat foods can actually contain more calories than the fat-containing equivalent because of the presence of this complex polysaccharide that's substituting for the fat. It actually brings more calories than the original fat. So sometimes it counts to pay attention to the ingredients. One of the things that we also start to notice when we start looking at the algae, and particularly the multicellular algae, is an idea known as the alternation of generations that's seen in the life cycles of multicellular algae, and also, as you're going to see starting next week, in plant life cycles as well. And this is one of the things that often confuses Biology 2 students a lot, this idea of alternation of generations. What do we really mean by this? Well, everyone knows that in sexual reproduction, there's always a haploid stage and a diploid stage. The problem is that when we think about that notion, we usually tie it to what we know about animal reproduction. So in animal reproduction, the haploid stage is the egg and the sperm. It is unicellular, a single haploid cell. And the diploid stage, well, that's the animal itself, you and me and every other animal. But in plants, the situation is different. And to some degree, it's also different in algae. And in fact, in Bio 2, one of the toughest things to wrap your head around is to recognize that the animal life cycle is a poor comparative tool for the life cycles of other organisms. Most other types of organisms, particularly fungi and plants, have reproductive life cycles that are very similar to each other and are very different from the animal life cycle. Yet our focus has usually been the animal life cycle. So here is the first idea that comes out of this notion that we can't use the animal life cycle as our measuring stick. And it's this idea of alternation of generations. Because what is meant here is that there is a multicellular diploid stage, no problem there, but there is also a multicellular haploid stage. Not a unicellular haploid stage, but a multicellular haploid stage. And it's only with that in the life cycle that you have true alternation of generations. And the algae is where we first encounter this. And you're going to see it in the plant kingdom next week, in the course of the next 2 weeks, because we have 2 chapters covering the plant kingdom, in the next 2 weeks. And there you're going to see alternation of generations yet again in all the life cycles of all the plants. It is a characteristic of plants, in fact. It's a defining characteristic of plants. And in many multicellular algae, we see the same thing. Now, in some multicellular algae, the diploid stage and the haploid stage are different from each other. And you can identify the 2 different stages. And we say that that is a heteromorphic life cycle. Hetero, different. So a heteromorphic life cycle has 2 different stages, one representing the haploid multicellular stage, one representing the diploid multicellular stage. But in some other algae, and you'll see an example a little bit later, the diploid and haploid stage look absolutely identical. You can't look at them and pick the difference. You have to use a microscope and look at the reproductive structures within the blades of the algae, the leaf-like features, to identify whether you're looking at the diploid stage or the haploid stage. And there we say they're isomorphic, remembering the prefix iso means the same. So if the 2 generations are different, it's heteromorphic. If the 2 generations are identical in appearance, then it's isomorphic. So here's an example of a heteromorphic brown algae. This is laminaria. So here is the adult diploid stage. And here is the multicellular haploid stage for the female and the male form. And you can see that they are distinctly different from the adult diploid stage. otherwise known as a sporophyte. Why a sporophyte, you may ask? Because you're going to ask the same question when we do plants. Because a sporophyte produces spores by meiosis, not gametes. Spores by meiosis. The gametes come from this multicellular haploid stage called a gametophyte. The gametophyte forms the gametes, the eggs and the sperm. Yes, I know that's very different from what you learned in Bio 1. This is something that you're going to have to get used to. Because as I said, in the plant kingdom and in the fungal kingdom, this is very characteristic. Meiosis produces spores. Gametes are the product of some other function, not meiosis. I'll remind you of that again when we do the plant kingdom in the next couple of weeks. Then we have the oomycetes. These have been classically characterized as fungi, hence the mycete part of their name. If you see mycete or mycota, you can be pretty confident that that's something relating to fungi. And so oomycetes have been historically classified as fungi. And in fact, if you do a botany class, because fungi were traditionally labeled as plants, you often cover the fungi, including the oomycetes. But oomycetes are now considered to be protists. They just happen to have an appearance that looks very much like a fungi, and that's because their life cycle and their behavior is very fungal-like. So they feed by phagocytosis and absorption. And in order to maximize phagocytosis and absorption, they produce tiny little filaments referred to as hyphae. The same name is given to the little filaments the fungi produce. So the naming conventions that are used are very much fungi because they were once considered to be fungi. The reason why they're no longer considered to be fungi is because in classical fungal taxonomy, fungi were always characterized as not having a flagellated stage, which oomycetes do. They have a biflagellated zoocyte, a reproductive stage, which is kind of ironic because when we get to the fungal kingdom, you're going to see that the clade of fungi that is considered to be most basal to the fungal kingdom now actually has a flagellated stage. But by genetic analysis, it is actually considered to be a true fungus. So, one of the original classically identified fungi is now a protist, and one of the classically identified groups that was once a protist is now a fungus. Yeah, isn't taxonomy grand? But that's the way it works. So here's the life cycle of a oomycete, and again, it's very fungal. Fungi do have both a sexual and an asexual reproductive strategy. In the asexual reproductive strategies, specialized hyphae do directly yield the zoospores that then germinate to give rise to the next fungus. However, in fungi, it is a haploid structure, not a diploid structure. So what separates the oomycetes from classic fungi is that asexual reproduction involves diploid structures, not haploid structures. If they're reproducing asexually, then you have two separate hyphae that produce the sperm and the eggs. They grow together, and the sperm then penetrate into the egg-carrying structure and fertilize the eggs, giving rise to the zygotes. Those then emerge and germinate to give rise to the next generation of oomycetes. And here you can see a fish growing on agar, or a dead fish laid out on agar, and all these little fibers are actually the hyphae of an oomycete breaking down the fish. doing exactly what a fungus would do. So one of the impacts of oomycetes is that they are major parasites of economically important plants. So Phytophthora infestans is what gave rise to the potato blight in Ireland and elsewhere around the world. So if you have Irish ancestry, it is almost certain that your Irish ancestors migrated here to America in response to the potato blight. About 1 million to maybe 2 million Irish died because of the potato blight from lack of food, but many more millions migrated to the United States, to Australia, to New Zealand, primarily to those countries to escape the potato blight. Phytophthora cinnamoni, unfortunately, was introduced into the United States on agricultural plants, and it turns out that it is a major problem for the American chestnut because the two did not co-evolve, and so it is killing the American chestnut. I know that the same oomycete was introduced into the state where I grew up in Australia, Western Australia, because someone thought it would be really great to start a cinnamon industry in Western Australia, because there are areas where the cinnamon trees can grow because it's conducive to their growth. What they didn't realize is that cinnamoni is hosted by the cinnamon tree because they've co-evolved. And so when it was introduced into Western Australia, cinnamoni got in amongst very important trees that were the heart of the timber industry in southwest Western Australia, and very quickly killed off a couple, or nearly killed off, a couple of very, very important hardwood eucalypts that were the heart of the timber industry in southwest Western Australia. Fortunately, it was recognized relatively early, and the Australian government research organisation called the CSIRO grabbed some seedlings out of the forest that appeared to be resistant to cinnamoni and cloned them and produced thousands of clones of cinnamoni resistant trees that they replanted back into the forest, and slowly but surely these trees are regrowing. And these are very, very impressive trees. They can grow to 250 to 300 feet in height. They look very much like a smaller version of the giant sequoia, in that while they may be as tall, their trunks are nowhere near as large in diameter, but they have very few branches low down. All the branches are way up near the top of the tree, which means that you have a lot of wood in the trunk that contains no imperfections because there are no branches coming out of it. And it's a hardwood, which unfortunately means too that the trees are relatively slow growing. But the timber industry is recovering. It's taken about 30 to 35 years now, maybe a bit longer, to get it back to a place where they can start cutting trees that are economically valuable for cutting, but the industry is on the way out. Phytophthora ramorum is another one that was introduced here to the United States, again, through the nursery trade, and this one attacks live oaks and native oaks here in the United States. And there's a slide at the end of the presentation that illustrates the distribution of this and the effect that it's having on oaks around the United States. It's pretty bad. The second clade are the alveolates. These are so named because they have little sacs under the cell membrane. So here's the cell membrane. Here's the flagellum. Remember, the flagella are again an extension of the cytoskeleton. They are encapsulated within the cytoplasm of the cell. So there's the extension of the plasma membrane wrapping around and going out to encase the flagellum. But here are what we're interested in, these little sacs called alveoli. Again, there's been a lot of discussion as to what these are for. What we think they're responsible for is for osmoregulation, controlling the flow of water into and out of the cells. That seems to be what everyone has focused on as the possible role for these alveoli. The clades that are found here are the dinoflagellates, the apicomplexans, and then a diverse group of organisms called the ciliates. So we'll address each of them in turn. So the dinoflagellates are photosynthetic. Of the three clades, they are the photosynthetic clade within this group. They are red in color because of a red accessory pigment. These are the quote unquote algae that give rise to the algae blooms that occur off the coast of Florida and also in some cases in fresh water in the lakes and streams in Florida. These produce, I guess, toxins, neurotoxins that are secreted from the cell. Not all species make them, but there are several genera within this clade that produce these toxins to paralyze fish because they're actually predators. They will eat the skin off the fish, but in order to capture the fish, they need to paralyze them first, and they produce these neurotoxins. And the neurotoxins stun the fish to allow the cells to then start eating the skin, and that obviously kills the fish. If you are down on the coast when one of these blooms is taking place, don't be surprised if your lips start to tingle, because the neurotoxins are being carried in the sea spray, and when it lands on your skin, it creates a tingling effect. Fortunately, it's not in high concentration, so you won't be affected by it. But other mammals like the manatees and dolphins, if they swim into these blooms, they can be killed by these toxins. They're pretty potent. Their name comes from the fact that they have two flagella in between that sit in the groove between the two plates that forms the outer covering. This plate is kind of a paper-like material that covers the cells. And when these flagella are active, it causes the cell to spin round and round. And so the dino comes from Dionysus, a Greek god, the god of celebration and wine. And in the celebrations associated with this Greek god, the celebrants used to spin round and round in a circle with their arms out. And so when people were looking for a Greek or a Latin name for these organisms, obviously the presence of flagella, that's where you get the flagellate, and the dino refers to the fact that they spin when those flagella are active. So dinoflagellates. And here you can see a red tide bloom off the coast of Western Australia, the northern coast of Western Australia. And you can see that it has turned the ocean red in color. This is the dinoflagellate bloom. I do have it. I haven't identified it, but let me write here. I haven't written today for some reason. No reason to. So this is hysteria, the predatory. Dinoflagellate. Yeah, it's hard to imagine a single cell being called a predator. But in this case, it actively paralyzes its prey and then eats its prey, which results in the prey's death. And that's predatory, however you want to slice it and dice it. So these are single cells that are truly predatory. The next group of alveolates are strictly parasites. These are the apicomplexans. They're so named because at the apex of the cell, there are a complex of organelles that are used to penetrate into the tissues and cells of the hosts that form part of a very complex life cycle that often involves two hosts, a primary host and a secondary host. They also have a non-photosynthetic plastid, so it's referred to as an apicoplast. It's located in the apex. And the retention of this non-functional plastid has attracted the attention of people that are trying to eliminate these parasites, because it's amongst this group of parasites that you find the malaria parasite Plasmodium. Malaria is still a major public health concern. Some 200 million individuals are currently considered to be infected with malaria. It may be a little bit more than that, somewhere between 200 and 300 million, I think is the current estimate. And somewhere around 500,000 to 1 million individuals die of malaria every year. So it's a major public health concern. And the problem with malaria is that the infective cells, once they get into the body, have developed a very clever way of preventing the human immune system from attacking it. So right now, it's not possible to create a vaccine for malaria. And the way they do it is to capture cellular proteins from the host cells that they've infected and insert those into their own membranes so that they look to the immune system like host cells. So the immune system doesn't touch it. Fortunately, there are drugs that can eliminate malaria, but also, unfortunately, some of the drugs are losing their effect because the cells are becoming, or the parasites are becoming immune to some of the drugs. You can also control the Anopheles mosquito that transmits malaria, but again, unfortunately, populations of the mosquito are becoming immune to the insecticide as well. So malaria is unfortunately on the rise again, becoming a greater problem, not a less problem. These used to be known as sporozoans because the infectious cell that initially infects the primary host is called a sporozoite. So here's the life cycle. So humans are considered to be the primary host. The mosquito is considered to be the secondary host. So when a female mosquito bites, and it's only the female mosquitoes that bite, to suck blood because they want the proteins from the red blood cells in order to be able to produce their eggs. They use the proteins from human red blood cells to build their eggs. And so they need a blood meal in order to be able to do that. So when she bites, the sporozoites, which reside in the salivary glands of the mosquito, are injected into humans because the saliva is injected in to act as a coagulant to prevent the blood from clotting. And so once they're released into the bloodstream, the sporozoites travel in the bloodstream to the liver because all the blood passes through the liver to be filtered. The sporozoites then infect the liver cell and reproduce as merozoites. So this is a form of haploid reproduction. The merozoites are then the cells that burst out of the liver cells and infect red blood cells. And there they turn into, this so I can see, into the gametocytes, which are in turn going to produce the gametes once they re-enter the mosquito. And this runs through cycles. The different forms of Plasmodium, because there are several species of Plasmodium that cause malaria, have different cycles of rupturing liver cells and rupturing red blood cells. And particularly it's the rupture of the red blood cells that matters here, because you may be familiar with the idea that malaria sufferers go through cycles of fever and chills. The fever is associated with the infection of the red blood cells. The chills are associated with the lysis of the red blood cells. And each different species of Plasmodium has a different cycle of fever and chills associated with infection and bursting of the red blood cells. But they're all equally deadly if left untreated. When a female mosquito then bites and draws in blood from an infected individual, she will get some of these gametocytes. Once they enter into the gut of the mosquito, they produce the gametes, both male and female gametes. The male gametes then fuse with the female gametes to fertilize it, producing the zygote. Within the zygote, meiosis occurs, producing haploid sporozoites, which then migrate up into the salivary glands and reside in the salivary glands, waiting to be injected back into a new human host again. Very complex life cycle, but very effective nonetheless, and unfortunately, deadly to humans. Then we have the final group of alveolites, the ciliates. And what characterizes the ciliates is the presence of two nuclei called a macronucleus and a micronucleus. And as you might guess from the names, one is large, one is not. The small micronucleus has a single copy of the chromosomes. It's a single diploid nucleus, single copies of all of the chromosomes, and plays no role in the day-to-day maintenance of the cell. It's strictly for reproductive purposes. The large macronucleus contains multiple copies of the genome. So it is not diploid, it is multiple. I don't know what term you'd want to use, but it is very large and contains multiple copies of the genome. And it is responsible for day-to-day running of the cell. Consequently, the reproductive cycle or the life cycle of a ciliate, and we'll see it played out in paramecium, introduces us to two new terms. And those terms are plasmogamy, fusion of the cell or by fusion of fusion of cells by fusion of the cytoplasm, versus karyogamy, the fusion of the nucleus, independent of fusion of the cell. This is not the last time that we'll see these terms, but it is the first time that you will have heard them. And these are going to become terms that you're going to have to be familiar with over the course of a number of clades, but most notably the fungi in the future. So the reason why that it's important to recognize that is that in the ciliates, transfer of genetic material is completely independent of the reproductive process. So they exchange genetic material while fused together by plasmogamy. Once they've exchanged genetic material, then they separate, and each cell will then carry out the reproductive process independent of the other. And it's still quite complex. So here you can see the basic makeup of a paramecium, a typical ciliate. Got a feeding groove to capture food particles, contractile vacuoles to regulate water flow, food vacuoles that are used for digestion and excretion of waste products, and then the micronucleus and macronucleus, as I've explained, one for day-to-day maintenance of the cell, one for reproductive purposes. So let's take a look at the life cycle. So the two paramecium cells that are going to mate, and we don't recognize male and female, something that you're going to see introduced, and I might as well introduce you to these right now, some new terminology. So the compatible mates are typically described as two mating strains which are referred to as plus and minus. So we don't have male or female. So you're going to see this idea of the plus and minus mating strains very particularly in algae life cycles, but it's a similar sort of idea here. So we've got these two compatible paramecium, the two mating strains. They fuse by plasmogamy. It's not identified in the book as such, but that's what is happening here. It's fusion by plasmogamy. This triggers meiosis in the two cells, but meiosis only occurs in the micronucleus. So it undergoes meiosis, produces 4 micronuclei. Of those 4 micronuclei, 3 of them dissolve away, leaving one behind, which then replicates by mitosis to leave 2 identical haploid nuclei in each cell. They then exchange these haploid micronuclei so that each one now has a copy of a haploid nucleus from itself and 1 copy from its mating partner. Once that exchange has taken place, the 2 cells separate. And then each cell undergoes its own round of separate division. So the fusion of the micronuclei is karyogamy. So it's not identified as such in the book, but that's what this process is, and it's going to be called that in later chapters. So now we've got 2 distinctly different nuclei. We've got a macronucleus that is genetically identical to what has been in this cell all the time. But now we've got a diploid micronucleus that is a recombination of the 2 micronuclei from each of the 2 mating paramecium. This micronucleus now undergoes multiple replication to give rise, again, by mitosis, to give rise to a total of 8 identical micronuclei, and they're all haploid. All diploid. I think I may have said haploid, but they're all diploid. So these are 8 identical diploid micronuclei. 4 of them then undergo multiple rounds of continued DNA replication to give rise to 4 new macronuclei that are genetically identical to the 4 micronuclei. At the same time, the original macronucleus now disintegrates because it no longer has any function. At this point, then, the paramecium then undergoes 2 rounds of division to give rise to 4 identical cells, each containing 1 copy of the micronucleus and 1 copy of the macronucleus. And these 4 cells then go off and live an independent life cycle, mating as necessary in the future. So as you can see, this is a fairly complicated life cycle. It's one of the most complicated protist life cycles that we have. And all the ciliates follow a fairly similar sort of life cycle pattern. The last clade of the 3 major clades in the SAR supergroup are the rhizarians. These are the classic amoebas. Sorry, and these are not the classic amoebas. These are amoeboid cells, but they are shelled amoebas. So they're going to have some form of shell around the outside of them. And depending on what type of shell determines which of the 3 clades within rhizaria that they belong to. So the radiolarians have a hydrated silica shell. The forams have a calcium carbonate shell. And then the cercozoans have a rigid shell around the outside of them that is somewhat like a plant cell wall, but it's not made up of cellulose, but it serves a similar sort of function. So the radiolarians are both marine and freshwater protists. I'm not sure why I didn't change this to marine and freshwater, because they used to be split into 2 separate groups Radiolaria were marine protists, and then the other group were freshwater, but we now know that they actually represent a single clade, so they've all been recategorized as radiolarians. So I need to change that. That should say marine and freshwater protists. And the shell around the outside, which is always referred to as a test, is made out of this hydrated silica. There are small holes in it that allows them to send out extensions. So you can see the extensions here. These are referred to as pseudopodia, but they are thin pseudopodia. They feed by phagocytosis, just like a typical amoeba. And so the main body of the cell, the amoeboid cell, is here inside the shell, and you can see the outline of the silica shell here. Those are the radiolarians. The forams, or foraminifera, have a calcium carbonate shell. Again, it is punctured by holes that allow them to send out these thin pseudopodia that allow them to feed by phagocytosis. And you can see a typical test here. It looks like a small seashell, but it surrounds a single shell. What is most interesting is that 90% of all known foram species are only known from their fossils, which indicates that in the past, there have been a significantly greater diversity of forams than there is now. We recognize somewhere around 3,000 species of forams currently extant, but in the fossil record, we think there may be as many as 300,000 or more distinct species. These are marine species, and they're really important in establishing timelines in marine sedimentary deposits. Each time period had its, in marine sedimentary layers at least, had its own unique populations of forams. And so as you go from one sedimentary layer to the next, you can identify which sedimentary layer you're looking at because of the populations of forams that are present on the basis of the tests. They're calcium carbonate shells that are left behind. And these have been used to establish the relationships between marine sedimentary layers around the world. And these have been, this relationship, this layering, this timeline was built by oil industry geologists, because oil and gas that we use as fossil fuels are the product of the fossilization of marine algae primarily. And so they're going to be associated with marine sedimentary deposits. And so to figure out where you could expect to find them in the marine sedimentary layers, you need to know which layers you're looking at. And you can use the foram tests to tell you which layer you're looking at, because each one has its own characteristic forams. And so the oil geologists built this stratigraphy on the basis of foram tests. Paleontologists have simply utilized it as well, because it's a good tool, it's a good yardstick that is well documented and well characterized, thanks to the oil geologists' work. And so we are piggybacking on the back of their work to allow us to identify this marine sedimentary layers in different locations around the world. What is also pretty impressive is the amount of limestone that has been produced as a consequence of the compression of these foram layers. If you've ever been to the southeast coast of Great Britain, to the area of Dover, one of the things that that region is famous for is for the bright white chalk slash limestone cliffs that can be over 300 feet high, but that chalk material extends quite deep into the earth as well. Those are the compressed calcium carbonate tests of billions and trillions of forams that have lived in the past, that have been compressed down to form these chalk layers. And that's pretty impressive, considering that these are the shells of single-celled organisms that have been compressed to form these huge chalk deposits, these huge cliffs that are built from these huge chalk deposits. So here you can see some 4M tests in the sedimentary rock. Then you have the cercozoans, which are again amoeboid, but they also have flagella in some species. These are the so-called photosynthetic amoebas, and Polynella is the cercozoan that I was referring to at the beginning of the presentation, that is strictly photosynthetic and has a green organelle. It's not strictly referred to as a chloroplast because it's got other material present as well, but it has the remnants of what appears to be a cyanobacterial cell wall made out of peptidoglycan. Remember, a lot of bacteria use peptidoglycan as a component of the cell wall, and cyanobacteria actually do this. And so the photosynthetic structure, this chromatophore, has the remnant of what appears to be a cyanobacterial cell wall because it's got these peptidoglycan materials associated with it, which suggests that this may actually be evidence for a second primary endosymbiosis of a cyanobacterium rather than a secondary endosymbiosis of an algae, something that took place much more recently in evolutionary time, because in Polynella, there is still a remnant of the cytoplasm and a nuclear remnant of the, oh, sorry, I shouldn't say nuclear remnant. That's, again, there's so much of this material, I'm sorry. Nuclear remnant is in the other cercozoans, which we do know are the product of a secondary endosymbiosis. But in this case, it's a primary endosymbiosis, no nuclear material, but cellular material left behind nonetheless in the form of the cell wall. I'm somewhat embarrassed by going that route. I ought to know better. Yeah, but in the other cercozoans that are the product of a secondary endosymbiosis, and we know for sure they are the product of the secondary endosymbiosis, there is a nuclear remnant, a cytoplasmic remnant, plus the chloroplast. They are photosynthetic, but they can also be heterotrophic because they capture bacteria and other microorganisms, but they can also carry out photosynthesis. So we would describe them as being mixotrophic in much the same way as I described them at the beginning of the presentation. So here's the chromatophore. You can see that these cells are flagellated, as I mentioned. Single photosynthetic structure. You can't see the peptidoglycan, but we can detect it. It surrounds this structure. Oops, I forgot to delete this slide, because this is the slide you want to look at. So we're going to take a break. I think I need one. Don't know about you, but I think I need one. So we're through the second of the 4 supergroups. Yes, I know it only seems like we're halfway through, but the next supergroup is very quick. So we will take a break, and I'll meet you on the other side with the answers to these 2 concept check questions. Why do the forams have such a well-documented and preserved fossil record? And would you expect the plastid DNA of photosynthetic dinoflagellates and diatoms to be more like the nuclear DNA of plants or the genomal DNA of cyanobacteria? See you on the other side of the break with the answers.